A very big hello to all my wonderful PU2 as well as KCET champions. I hope all my dear children are doing absolutely good. So in today's video, we are going to deal with the chapter Molecular Basis of Inheritance. And before I go ahead, I want to share something very personal with you guys. So there is a thing that I have noticed, okay? Whenever something is paid, you guys ask that, sir, ma'am, can you make it free? Can you make it free? Okay. There was a time when we launched Sankalp series and then we shifted to midterm as well as all the examination that came in, and you asked, can you relaunch the Sankalp series? Okay, fine, we did it. But then the thing that we see is that whenever something is given to you for free, you guys are not making the best use of it. So every time Masterstroke, the first video is going to be released for the Masterstroke series, yes, there's a huge hype around it. Later on, it goes down. Is it because we do not teach well, or what is it about exactly? I really feel very sad because, you know, it's not very easy to come in front of the camera and to talk for like 2 hours straight without having anyone around you. You're just talking to yourself in a hope that this video which you are making will be useful for the children who are watching it. And if you guys are not making the best use of it, do you think it's justice? No. So we are also human beings, we also need motivation, and our motivation comes from you. When you listen to the lecture, when you do the homeworks, when you take things seriously, when you comment down that, yes, sir, ma'am, the lecture was good, or basically you ask us doubts, all of this is a mode of motivation, a mode of love that you can show to us. So my dear child, if at all you are watching this and if at all you think that whatever I said makes sense, definitely take it seriously. All this content that we are making is going to be useful for you. We are putting in a lot of our hard work and justice to it. Okay, we are not skipping out any topics out there. We are giving you each and every single bit of the chapter out there such that you do not miss out on any marks in your PU2 and KCET. So do not forget what I have told and take all the lectures seriously and watch it up. Okay? Fine. So now we'll study the molecular basis of inheritance, and the first thing that we have is this. What is this? This is the list of organisms with their genomic size being mentioned up over here. So you can get questions such as match the following. You have to match the genomic size. This question came in KCET. You can get questions in your PU2 also. They can ask you what is the genomic content of this particular organism. You should be knowing about that. So the genomic content is going to be the least in bacteriophage 5x174. 5x174, even though the name is really big, it has very less genomic size, that is 5386 nucleotides only. So ma'am, which organism has the highest? It will be obviously us, human beings. In human beings, my dear children, the genomic size is 6.6 into 10 to the power 9 base pairs. So ma'am, here also human being is written and here something else is given. What is that? So listen to me carefully. Normally a cell which is there, it has 46 number of chromosomes, right? Do you agree? 46 number of chromosomes are there. I call this condition as diploid. And this has been shown up by 2n, okay, double the number of chromosomes. Here the genomic content is going to be 6.6 into 10 to the power 9 base pairs. Now, when you talk about gametes such as ovum and sperm, both of them are having haploid content. The haploid content come together to form a diploid zygote out there, which is why over here, when I say it's haploid, that means it is half of it. Only 23 chromosomes are going to be there, and I'll call this as what? Haploid. And my dear children, this is denoted by just n. Half of 2n is equals to what? N. And over here, the genomic content also becomes half. It becomes 3.3 into 10 to the power 9 base pairs. This is how it is going to go. Got it? And now you can go questions like this. So what is the genomic size of a primary oocyte? So primary oocyte, my dear children, is just a normal cell which is diploid in nature. It has not started its division. So which is why it will be 6.6 into 10 to the power 9 base pairs. So ma'am, you said ovum. Ovum you have to form, no? Egg is there, that will have the haploid. As of now, this is the precursor of the egg. It has not completed the division. This is diploid in nature. Now, when it undergoes meiosis 1, it forms what? Your secondary oocyte. The secondary oocyte which is there, it will be having haploid. From this point onwards, okay, you get a polar body and you get a secondary oocyte, right? Again, when you divide, you get an ovum and then you get the secondary polar body. First the first polar body and the secondary polar body, and here it is the secondary oocyte, and this would be the ovum. Once meiosis 1 has ended up, the chromosome number has reduced, and it remains the same in the ovum also. So secondary oocyte will be having 3.3 into 10 to the power 9, and primary oocyte is not like that. It will be having 6.6 into 10 to the power 9 base pairs. Have that in your understanding. Cool. Now, you have two more organisms, bacteriophage lambda, okay, it has 48,502 base pairs, and we have another thing. So, both of these bacteriophages, they are not bacteria, they eat bacteria, they are actually viruses, okay, and now we'll talk about an actual bacteria, that is Escherichia coli, or you also call this as E. coli. E. coli has a genomic size of 4.6 into 10 to the power 6 base pairs. 4.6, 10 to the power 6. 4.6 into 10 to the power 6. Don't make it 10 to the power 9 and all. It will be very, very, very wrong. Okay. So 4.6 into 10 to the power 6 base pairs for E. coli in human. 6.6 into 10 to the power 9 base pairs. Okay. Fine. Works. So this would be done. Now, going ahead with what your DNA is going to be made. What do you think? Your DNA is made up of what all things? Your DNA is made up of a sugar. Okay, apart from that, you have a nitrogenous base, and apart from that, you have a phosphate group. So when sugar, just forget about this part. When sugar as well as nitrogenous base come together, you call this structure as nucleoside. Okay, fine. When sugar, nitrogenous base and phosphate comes together, you call this structure as what? Nucleotide. So nucleoside is different. It just has sugar and nitrogenous base. S, okay, starting with S. Sugar, nitrogenous base, it has nucleoside. Sugar, nitrogenous base and phosphate group, it forms what? Nucleotide out there, right? And before I go any further, I want you to understand the structure of all of it, which is why I'll go a bit forward and I'll try to make it over here. So, listen to me very carefully. Fine. Over here, I have a sugar. This sugar has 5 carbons. 1, 2, 3, 4, and the 5th carbon is right up over here. This is going to be a pentose sugar. Why? Because there are 5 carbons out there. On to the first carbon, you would be having, okay, just before I go ahead with that, I want to tell you one more very important thing. So this was a sugar, right? This was a sugar. Now, if at all, on the second position, you have OH group. If at all, on the second position, you have OH group, you will call this sugar as what? Ribose sugar. So, I'm just writing ribose will be having an OH group at what? The second position. But, if at all, I remove the O up there, if I am removing the O up here, what happens? Deoxygenation happens. So can I say this is going to be deoxyribose? Yes, deoxyribose. And I'm not showing that, okay? I am not showing that. I'm just writing it over here. Deoxyribose do not have OH group at the second position. Just H will be there, O will be removed. Fine. Now, okay. The first carbon of this particular deoxyribose in your DNA. Your DNA is made up of deoxyribose. Your RNA is going to be made of ribose. That is how it goes. And on the first carbon, you have a nitrogenous base being attached. This bond is known as N-glycosidic bond. What do you call this? N-glycosidic bond is out there between the first carbon and the nitrogenous base. At this point of time, what do you call this? Nucleoside. Now what happens is that basically on the 5th carbon, a phosphate group comes and attaches. On the 5th carbon, a phosphate group comes and attaches. What do you call this bond as? There is a carbon over here and it is bonding with phosphate, and you need to understand that in between that there is oxygen also. Anyways, if at all you do not remember that, it's okay. But just understand that carbon and phosphate is being bonded over here through oxygen, and which is why you will call this bond as phosphoester bond. So you have N-glycosidic bond and you have phosphoester bond right up over here. This whole structure is what? Nucleotide. What is it? Nucleotide, because you have everything together. Now, okay, I got the nucleotide. How do you make the DNA then? Let's see that. So over here, I have one deoxyribose, the nitrogenous base right up over here, and the phosphate group over here. This is how it goes, right? So I'll just make some changes. This is your 5th carbon. 1st, 2nd, 3rd, 4th and 5th carbon, and over here you have the phosphate group being attached. So I'll tell you one very important thing. Whenever you are going to make the nucleotide, it's not like the phosphate group alone comes and attaches over here. No. Actually, it is a structure which have 3 phosphate groups. That means you call this as triphosphate. Okay, triphosphate. The whole thing is known as DNTPs. Deoxynucleotide triphosphate. Deoxynucleotide triphosphate. Now this is up there. Similarly, you will have another nucleotide structure just like this. First carbon, you have nitrogenous base. Second carbon, third carbon, fourth carbon, fifth carbon, and here you have the phosphate group, right, on the 5th position. Now what happens is that here you have a carbon, and this carbon also makes a bond with this particular phosphate group. So here also carbon made a bond with phosphate group. Here also carbon made a bond with what? Phosphate group. That's when there is a division over here. There's a breakdown of bond over here. These 2 phosphates are going to separate, and this phosphate guy is going to go up, team with them. Again, since it is between carbon and phosphate and it is also through oxygen, I will call this bond as what? Phosphoester bond. So here also phosphoester bond, here also phosphoester bond. Phosphoester, phosphoester. Can I say from the 3rd to 5th carbon, from the 3rd carbon of one nucleotide to the 5th carbon of another nucleotide, you have basically what? Phosphodiester bond. Yeah, there are 2 ester bonds out there. So I will call this now as phosphodiester bond. Now you need to understand one very important thing. They have bonded. Suppose the strand is like this. Over here there is nothing else to bind. So I will call this end as the 5' end. Over here also I have nothing else to bond. So basically I will say it is the 3' end. So you might have seen in DNA we have 5' end and we have 3' end. That is what I mean by this. And similarly you will be having one on this side also. But it is going to be ulta. That means for the other one over here, you will be having the 3' end right over here and 5' end right over here. Why? Because both of them, if at all it looks like this, the nitrogenous bases are going to go on that particular side. So here also nitrogen base this side and there also nitrogen base that side. No. Actually, nitrogenous bases which are there, they should be from that side and this side, both of them should be coming inside and should be making bonds between them. That is how you have the 2 strands of DNA being bonded. So can I say that this is an anti-parallel structure? Yes, they are parallel, but they are ulta also. So I'll call this as what? Anti-parallel structure. Got it? Yes. And in between the nitrogenous bonds out there, you have what? You have, sorry, nitrogen bases out there. What do you have? You have hydrogen bonds. What bonds? Hydrogen bonds. Listen to me. Nitrogenous bases which are there, they are also of 2 very different types. One is purines and the other one is pyrimidines. The purines which are there, you know, there are people who call AG, OG, right? Similarly over here you have AG, adenine and guanine. They are what? Purines. In pyrimidines you have what? You have thymine as well as cytosine. In between adenine and thymine, you have 2 hydrogen bonds, thymine T and 2 also T. So basically 2 hydrogen bonds are there. G and C, since they look very similar, almost very similar, they are more closely linked. They have 3 hydrogen bonds. That's how it goes. Now this is the case of DNA. In the case of an RNA, you didn't have thymine. Instead, you have something else that is known as uracil. In the case of in RNA, you do not have thymine, you have uracil. Uracil base pairs with adenine. So RNA and DNA, I got to understand 2 very different things out there. In RNA, there is ribose sugar. In the 2nd position, you have an OH group. In DNA, you have deoxyribose sugar where you do not have the O up there, right? Similarly, in the case of a DNA, you have A and T being bonded, that is adenine and thymine. But in the case of RNA, what do you have bonded? You have A and U, that is uracil being bonded. Okay? Okay, so out of them, who came first, ma'am? It was uracil which came first. And in the uracil, if at all you make certain changes, if at all you add a methyl group at the 5th position of uracil, you get thymine. So from this guy came who? Thymine. Okay. So this is the basic thing, which is what you need to remember. Now, let's go back. Let's go back and see what is up over here. So nucleoside formation, a nitrogenous base links to the first carbon of the sugar. Yes. What do you call this bond? N-glycosidic bond. Okay. In RNA, you will now call this structure as adenosine, guanosine, cytidine and uridine. Okay, when nitrogenous base and the sugar, that is the ribose sugar, comes together, you will call this adenosine, guanosine, cytidine and uridine. But in the case of DNA, what do you need to do? You need to add the term deoxy. Okay, so deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. This is what the nucleoside structure. Again, they can ask you, what is the DNA being formed of? Adenosine plus triphosphates or something else. So you should know its DNA cannot be made of adenosine. It should be deoxyadenosine. Got it? Yes. Okay, fine. Now nucleotide formation. A phosphate group attaches to the fifth carbon of the nucleoside via the phosphate bond. Yes, we know that phosphoester bond, right? Phosphoester bond. And now you have two of them, you call them phosphodiester bond. Okay. Nitrogenous bases, if at all you see, you have purines, adenine and guanine. Pyrimidines, you have cytosine as well as thymine. In the case of RNA, it is uracil. Pentose sugar which is there, it is deoxyribose in DNA and ribose in the case of RNA. And then you have a phosphate group also. RNA versus DNA. RNA has an additional OH at the second carbon of the ribose and RNA contains uracil instead of what? Thymine. And thymine is also known as 5-methylurazine. This particular question is also important. I think this was there in your model question paper this year. So in the uracil component, if at all on the fifth position you add a methyl group, it becomes what? Thymine. So 5-methyluracil is for thymine. Got it? Yes, ma'am. So until now, whatever we studied, that is being done. Now, polynucleotide chain formation. The nucleotides comes together, stacks up together and it forms a chain. Nucleotides link via 3,5 phosphodiester bonds. 3,5 phosphodiester bonds. Yes, correct ma'am. The chain has a 5' end free phosphate group and a 3' end which will be having free OH group. So basically at the 3' position, my dear children, this particular carbon is actually linked to a OH group. That is why I told you in between the carbon and phosphate you have an OH, okay, or an oxygen out there. You see, this is how it comes. It's not like plain phosphate is gonna come. It's not like that. Okay, the phosphate is actually attached to an OH group also. Okay, so yes, works. So I hope that is crystal clear. Fine. So on the third carbon, you have the OH group and that OH group attaches with the phosphate right there. If at all the phosphate is not there, the OH group remains free, which is why you call this as 3' OH end. Okay, fine, done? Yes. So 3' end is the free OH group and the backbone which is there is formed by sugar phosphate units with nitrogenous bases projecting inward. So basically backbone of DNA, if at all you are asked, it is sugar. It is the sugar and the phosphate. They form the backbone. Cool? Yes. Now, if at all you are asked about the structure of DNA, how do you define it? Two anti-parallel polynucleotide chains, one from 5' to 3' end goes and one goes from 3' to 5' end with a sugar phosphate backbone and bases projecting inwards. The bases which are there, they are projecting what? Inwards. Next thing, base pairing via the hydrogen bonds. A and T has 2 hydrogen bonds, G and C has 3 hydrogen bonds. Okay. Now, right-handed helix is there. It turns in a right-handed format and my dear children, the pitch of, okay, with a pitch of 3.4 nanometer. What by that you mean this particular distance that is there, this is known as a pitch. Okay, one whole helix is known as a pitch. The pitch of this particular thing is 3.4 nanometer and in a single helix, you know what, how many base pairs are going to be there? In a single helix you will be having 10 base pairs, 10 base pairs, and in between that the space between each base pair units out there, if at all you are asked, it will be what? 0.34 nanometer. So just divide it and you will get the answer. Okay, so whole length is 3.4 nanometer. You have 10 base pairs. If at all you want to know the distance between them, 3.4 divided by 10, it will be 0.34 nanometers out there. So that is exactly given over here. Right-handed helix with a pitch of 3.4 nanometer, 10 base pairs per turn and 0.34 nanometer per base. Okay, now base pair stacking and hydrogen bonds provide structural stability. The base pairs which are stacked together and also the hydrogen bonds provide structural stability to the entire DNA structure. Got it? Yes. Okay. Let's go. Now, everyone who is out there who is preparing for KCET, we have launched the ABHYAS KCET test series where there are 35 mock tests, there are chapter-wise PYQs, and all of it under perfect timed condition. So, if at all you are someone who is preparing for KCET and needs a resource, this is going to be a perfect resource for you because this was trusted by most of the children the previous year. They got the best benefit of it. We got ranks like 171, 443, 693, and the list is going on like that, right? So obviously this is something that you can take up, and as of now I will not recommend you to go ahead with the mock test, but I'll definitely request you to please solve the KCET PYQs once you're done with the chapter. You do not have any separate times to do all of the things together. You also have to cover the PU1 post your examinations are done, right? And the pricing for this is rupees 1499. If at all you want, you can check it out. The link will be given in the description below. Okay, now the discoveries which are there. Who discovered what? That is also important. So, in 1969, Friedrich Mischer, he was a Swiss physician out there, and he said that basically inside the nucleus there is some acidic substance. Okay, acidic substance, and he called this structure as nuclein. He did not call it DNA. He did not know about it. He said that there is something acidic inside the nucleus, and that structure is something that I call as nuclein. Who was this person? Friedrich Mischer. Who was he? A Swiss physician. Okay. Now in 1953, so many years after that, James Watson and Francis Crick came into the picture, and they said that there is a structure that is DNA, and it has what? Double helix model. And they could not do it alone. They took the help of the data from Maurice Wilkins and Rosalind Franklin's X-ray diffraction data. So James Watson and Francis Crick came together to give the double helix model of DNA, and they took the data from X-ray diffraction method or the data from Maurice Wilkins as well as Rosalind Franklin out there. Okay, now going ahead, base pairing. Who gave an idea about base pairing, that nitrogen bases are going to base pair between each other? It is Erwin Chargaff who gave an idea of the base pairing. He said that the ratios of adenine to thymine and guanine to cytosine are constant and equal to 1. Adenine to thymine ratio as well as guanine to cytosine ratio is equals to 1. Why? Because how many number of adenine is going to be there in a double-stranded DNA, that many number of thymine will also be there. How many number of guanine is going to be there, that many number of cytosine is also going to be there. If at all you're talking about what? A double-stranded DNA structure. Okay, so this was given by Erwin Chargaff. Okay, Friedrich Mischer, acidic substance, nuclein. James Watson and Francis Crick, the DNA double helix model by the X-ray diffraction data by Maurice Wilkins and Rosalind Franklin. Okay, and then you have Erwin Chargaff who gave the idea about the base pairing as well. Now going ahead, you have one very important thing. What do you think is the dimension of the nucleus out there? The dimension of the nucleus is, my dear children, it's just 10 to the raised to the power minus 6 meters long. 10 raised to the power minus 6 meters long. That is your nucleus dimension. And how much DNA do we have? We have a genomic size of 6.6 into 10 to the power 9 base pairs, and in between each base pairs we just studied 0.34 nanometer space is there, and if at all you convert into meters, you will get something like this, that is 2.2 meters length. So 2.2 meter length DNA, how do you think that they can fit into 10 raised to the power minus 6 meter long nucleus? Obviously very difficult, right? So which is why you will need the help of what? Histone proteins. You will need the help of some proteins which will be helping us in packing of the DNA. And before I go any step ahead, I want to tell you one very important thing. This is found in the case of eukaryotes. What are eukaryotes? Eukaryotes are organisms who have cells, cell membranes and everything out there, and they have organelles which are basically, just a second, okay? They have organelles which are having membrane around them. They have membrane bound organelles. Their nucleus have a specific region. On the other hand, you have some organisms known as prokaryotes. You know how they are? They do not have any nucleus. Their genetic material is going to lie up at a location, and you call this location as what? Nucleoid. But they do not have a specific location, so that there is no policies that you have to fit in the whole genetic material inside a nucleus, because there is no nucleus at all. So do they need histone proteins? The answer is no. Do we need histone proteins? The answer is yes, because we need to fit it inside the nucleus. And also one more very important thing, their genomic size is very less in comparison to us. We studied, it was in E. coli, it was 4.6 into 10 to the power 6 base pairs. But over here, is it like that? Okay, it is 6.6 × 10 to the power 9 base pairs. Okay, so those are the two things that you need to keep in mind. What are two things? That prokaryotes do not have histone proteins. Why? Because they do not have a specific region for packing their DNA material or genetic material out there. It just stays there, they do not have nucleus. Second thing, the genomic size is also less. Okay, now when we talk about histone proteins and their packing, you need to understand one very important thing, and that is this particular structure. What is this particular structure? Let's decode it. So basically there are almost 8 histone proteins which comes together. 4 in front and 4 in back. How many? 8 of them. So I will call this structure as histone octamer. I will call this structure as histone octamer. And onto this histone octamer, your DNA is wrapped around. How many times? Twice. Twice your DNA will be wrapping around. When I say it wraps around twice, I say that almost up to 200 base pairs are being covered here itself. In one particular histone protein, 200 base pairs are being covered. Is this important thing, ma'am? Yes, the answer is yes. You have to remember 200 base pairs. You also have to remember that it turns around twice. Now, the second thing that is important is that you do not just have these histone proteins. You have one more very important histone protein, and that is specific, that is different. You call this as H1 histone. You call this as H1 histone. And now the whole structure which is there, this nucleosome plus, this particular histone octamer plus the H1 histone, you will now call this structure as what? Nucleosome. Nucleosome. And you need to understand that over here, there would be multiple nucleosomes which are there. Multiple nucleosomes. So if at all you see, your DNA is going to wrap around these things, and it looks like on a string there are several beads. On a string there are several beads. So all these nucleosomes, when they come together, when you have a bead on a string like structure being formed, you call this as chromatin. Chromatin. And the chromatin, when it condenses even more, when they come close even more, you will now call this structure as what? Chromosome. That is how your chromosome looks like this, because you have condensed chromatin out there, and the chromatin in turn is made up of these multiple nucleosomes. Now there might be one important question, that is, ma'am, how does DNA wrap around the histone proteins? Is there something in the histone which makes it wrap? The answer is yes. We studied, Frederick Mishra told that DNA is what? Acidic. And when we say something is acidic, that means it is negatively charged. Dominantly, it is what? Negatively charged. And over here is the phosphate group which has the negative charge out there in the DNA, right? Now, what if the histone proteins are made up of, they are proteins, right? And proteins are made up of amino acid. Amino acid. And I tell you that these amino acids are predominantly the positive ones. So if we have positive amino acids, the negatively charged DNA can wrap around it. So what are these positively charged amino acids which are dominant in the case of histone proteins? My dear children, you have to remember their name, and the name goes like this. It is arginine and lysine. AL, arginine and lysine. These two histone proteins are there which are, histone amino acids are there which are super important to make the histone proteins highly positive, such that the DNA can easily wrap around it. Got it? Yes. Okay. So basically what did we study? That this is a nucleosome. This whole thing is a nucleosome, and it is made up of histone octamer. Apart from that, you also have what? H1 histone out there. And the DNA wraps around twice. By twice I mean 200 base pairs are being covered. Why does it wrap around? Because DNA is negatively charged, and your histone proteins have amino acids which are positively charged. For example, arginine and lysine. And H1 protein is between the two nucleosomes out there. H1 protein will be there between the two nucleosomes. That is how it goes. Right? Now, one more very important thing. I told that this structure which is being formed where beads on a string like thing is there, you call this as what? You call this as chromatin. And you can clearly see this. This is what the chromatin out there. And the chromatin will later condense to form the chromosomes, as I told you. The chromatin which is there, it is of two different types. It is of two different types. One is euchromatin, and the other one that we have is heterochromatin. And this is very easy to imagine. Euchromatin, Baba, it looks like this. You have your H1 histone, H1 histone, H1 histone, right? So if at all you see this, and I'll give you a comparison with the other one. Okay. if you compare this and this, what do you get to understand? That they have much space between them and they are very densely packed, right? So this structure, which is little loosely packed, is known as what? Euchromatin. And since it is loosely packed, it will take up stain very lightly. So it is going to stain very lightly. Whereas on the other hand, the next thing, that is heterochromatin, my dear children, it is going to be densely packed and they take up dark stains. Okay, or basically I can say that they stain really dark. Under the microscope, if at all you see, they look really dark. Now one more difference that we have between them and them is that here, since you have space, and this is your DNA, you know, right, the DNA which is there gives information to form a protein. How does it do that? So DNA which is there, it undergoes a process which is known as transcription. It makes an RNA, and this RNA undergoes another process that is known as translation, and finally you get a protein. So for this process to happen, transcription to happen, you need certain enzymes to come and attach over here and go ahead with the formation of an RNA. And for that you need space, and that space is provided over here, which is why I say euchromatin is transcriptionally active. That means transcription process can smoothly occur over there. On the other hand, heterochromatin which is there, it is transcriptionally inactive chromatin. It is not very active, it is going to be inactive chromatin. Got it? Transcriptionally inactive, because there is no space for the enzyme to come and attach over here. Fine, that's all. So you might be asked this question in your PU2, what is the difference between euchromatin and heterochromatin? Please know about it. And also remember, it's not just histone proteins which are there to pack the DNA. There are some non-histone chromosomal proteins also which are there in the chromatin formation, which are required in the chromatin formation. I hope that is crystal clear now. Yes? So that is how it goes. I hope that is being done. Now we go ahead with the search of genetic material. You know what? So yes, it was said that yeah, there is nuclein, there is this, there is that, DNA is double helical structure and blah 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 blah. But people were not able to identify what exactly is the genetic material, what is being passed down from one organism to the other, and how do we look almost similar. Our parents and us have some similarity. Obviously, there has to be some reason. Something is getting passed on. And then came the search of genetic material when there was so much confusion around it. And the first guy to come into the picture was Frederick Griffith. Not Frederick Misher, this is Frederick Griffith. So you can also remember, there also the first guy was Frederick Misher. Here also there is Frederick only, but the name is Frederick Griffith, and this happened in the year 1928. 1928. My dear champions, do you remember this year? It was 1869. It was 1869 when Frederick Misher came up saying that there is something. Then later on, in this particular time, that is in the year 1928, you had Frederick Griffith coming into the picture. 1928, and after that only all of this thing happened. DNA double helix structure and things came into the picture. Anyways, let's go back. We were right over here. Yes. So what exactly happened over here? Let's see that. So to check what's the genetic material, Griffith chose a bacteria that is Streptococcus pneumoniae. Streptococcus pneumoniae, my dear children, causes pneumonia, right? Now, there are two strains of Streptococcus pneumoniae. One is the rough strain which is there, and one is going to be the R strain. Sorry, one is going to be rough strain, that is R strain, and one is going to be the smooth strain, that is known as S strain. Why is it smooth? Because this particular strain has one extra covering around it, a smooth covering around it, which is made up of polysaccharide. So this one has an extra covering around it, and it is made up of mucus polysaccharide. Polysaccharide is nothing but carbohydrates. Yeah, that is why it gives a smooth appearance under the microscope. Whereas when you talk about the rough strain, it does not have it. Okay, is that a big problem? Is that a big deal out there? Yes. The rough strain, since it does not have the, what do you call, the coat around it, it is definitely going to be what? Non-virulent. It does not cause any trouble. If at all a person is infected with it, they are not gonna die. But on the other hand, the smooth strain which is there, it is the virulent form of the bacteria. It is the virulent form of the bacteria killing up the bacteria. That's a big problem. Okay, so then he took mice. I call it Puchu, my children know that, yeah. So Puchu was there, my dear children, and Puchu was given live R strain. That means living R strain. Now R strain, you know, it's rough strain, Papa guy, it's Papa, name is rough, it is not actually rough. It's cool, okay, does not cause any trouble, the mice will be living, no problem. Now he put live S strain into the mice. You know what happened? The mice died. Why? Because it was the virulent one. He got to understand that, okay. Then what did he do? He treated the S strain with some heat. He treated the S strain with some heat, and then okay, and then he put it in the mice. You know what happened? Mouse lived. It didn't die, it lived. Okay, so Puchu was really happy. After that, he thought that, okay, this and this is not making the mice die, so why not combine it and try to put it? Let's see what happens. So he did that, and you know what happened? Puchu died. Puchu died, my dear children, and not just that. Inside Puchu, he found live S strain. Did he ever put live S strain? No. He put heat-killed S strain and he put live R strain. He never put live S strain. From where did he get the live S strain? Inside Puchu, inside dead Puchu. Probably that means that from here, from the heat-killed S strain, something is passed into the live R strain, transforming this R strain into what? A S strain out there. That might be happening. So with that information in his head, he came up with this, some transforming principle, something, okay, transferred from the heat-killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. So basically something passed from here to here, which made the R strain make the coat around it and transform into what? The S strain. Now this thing that was getting transferred is known as what? The transforming principle, which is why the Frederick Griffith experiment is also known as the transforming principle experiment. That is how it goes, right? Now, let's go ahead. Let's talk about the next thing. So obviously there was a huge lot of confusion. Griffith did not have any idea to it, which is why three other scientists came into the picture, and that was Oswald Avery, Colin MacLeod and Maclean McCarty. So Avery, MacLeod and McCarty, you can just call them like that, and they came in the year 1933 to 44, just after 1928, and then after a few years they had come into the picture, not after a few years, very long years. Anyways, so they came into the picture and they said, okay, so you did the experiment, let's see what we can do about it. So they took the heat-killed S cells, okay, the heat-killed S cells. And you know the heat-killed S cells will be having DNA, it will be having RNA, it will be having protein. And now we have to confirm what exactly is the thing that is going to, is responsible for transformation. Is it DNA, is it RNA, or is it the protein? So what they did is that, they took the heat-killed S cells and they added proteinase into it. That means now the heat-killed S cells is not going to have the proteins. Why? Because the proteinase is going to cleave the proteins down. Okay, no proteins is there in the heat-killed S cells. Now in this condition, if at all I mix the heat-killed S cells and R cells, do you think transformation is happening? Yes, it happened. S cells still appeared. That means something was still there which was not protein, which was getting transferred, because if it was protein, the transformation could not have taken place because there is no protein at all. You have killed you have broke down all the proteins out there. So this didn't work. Okay. Now what about adding some RNAs that is going to cleave down the RNA molecule out there? They did that also. Still they found what? S cells were appearing. That means still transformation was taking place. Still there is something that is responsible for transferring some very important thing out there. Then they treated it with DNAs, that is going to cleave down the DNA. And you know what? What happened? No S cells appeared over here. That means probably it was DNA which was passed from the heat-killed S cells to the R cells transforming into S cells. Now since DNA is not there, this transformation is not taking place. So probably DNA is the genetic material. So they came up with this conclusion. Avery, MacLeod and McCarty, they came up with this conclusion that DNA is definitely the genetic, so did I say it's not the genetic material? I'm so sorry if I said that. But anyways, DNA is the genetic material. They came to a conclusion like that. But you know what? People didn't believe them. Which is why one more experiment was needed. And that was the last experiment, and this is one of the famous experiment done in the year 1952. So if we go back and if at all we see over here, 1953 you got the DNA double helix. Just before that you got to an understanding that yes, DNA is definitely the genetic material with the help of the Hershey and Martha Chase experiment. Okay, Alfred Hershey and Martha Chase. So over here finally proved that DNA is the genetic material, but how did they prove? That is also a question. So my dear children, listen to me carefully. over here, they took the help of two organisms. One is E. coli and the other one is bacteriophage. Bacteriophage, as I told you, it's a virus that feeds on bacteria. So bacteriophage is there. Now they took two things, radioactive sulfur and radioactive phosphorus. Radioactive sulfur and radioactive phosphorus. Okay. It was understood that phosphorus is a part of DNA. Your DNA is having phosphate group, right? So obviously phosphorus is a part of your DNA. Whereas sulfur is a part of what? The protein which is there. Cool? Yes. So over here, this particular coat of the bacteriophage, okay, or basically I can say wherever the protein is there, that is going to be tagged by the radioactive sulfur. What isotope was used? It was S35 isotope which was being used. Over here, suppose we already know that your DNA is made up of phosphate, which is why we know that only the DNA is going to be radioactive over here. Rest is not. Anyways, so basically we know that DNA is radioactive over here, and we also know that protein is radioactive over here. They also knew that. Yeah, they also knew that, right? Now, they treated it with E. coli. Similarly, with E. coli, what is going to happen? So from here, whatever the genetic material is, it can be protein, it can be DNA, definitely that has to be passed into the E. coli. Why? Because when it is passed into the E. coli, that genetic material is going to incorporate into E. coli's genetic material and form all the things which is needed by the virus, and later form the viral babies out there. That is the basic logic out there. So they thought, okay, let's infect it. Anyway, the genetic material is going to pass inside. Okay, the same thing was being done here, same thing was being done here. So first is infection. Second one we did is blending. So basically, we blended it such that anything which is there on the surface can be moving apart from it. So that is known as blending. Now the third thing that we have is centrifugation. Whatever is there, we are spinning it up, making sure that there is a precipitate out there, and everything which is heavy will be coming down and settling down, and whichever thing is light, it will be just at the top. So this thing which was at the top, it is known as, just a second. So over here, yes, over here you had two things. One is your supernatant and one is going to be the pellet. Similarly, the supernatant and the pellet. Pellet is going to come reside down and the supernatant stays like that. Okay, I'll write that also, supernatant, and this is what? The pellet. In this particular condition, and my dear children, you only think what is going to settle down. Obviously the E. coli and everything is going to settle down because that is more heavy, right? So E. coli is going to settle down definitely. So over here they found that radioactivity was not found in the E. coli. Okay, where was it found? In the supernatant. In the supernatant they found the radioactivity. Okay, what about here? Here they found radioactivity in the pellet and not in the supernatant. That means what? If it is there in the pellet, that means whatever the thing was there, it passed to the E. coli. And what was the thing that was getting passed to the E. coli? It was the radioactive phosphorus. And radioactive phosphorus, we all know, it was labeling what? The DNA. So probably the DNA is something that is passed on to the E. coli, which is why there is radioactivity seen in the pellet. Whereas over here, the proteins are not passed into the E. coli, which is why there was no protein here. Okay, no radioactive protein over here, but it was still there in the supernatant, because in the supernatant you had the bacteriophage and everything out there. You get it? So that is what confirmed that DNA is the genetic material with this particular experiment. Always remember, it was P32 and S35. So 5 looks like S only, so you can remember like that. For S35 and P32, that was used for this particular experiment. Okay, done. That's all. This is Hershey-Chase experiment, and with that people got an understanding that what is the genetic material as well. Now, going ahead, the next topic, what is the first genetic material? It was definitely RNA. It is not DNA, it is definitely RNA. Okay, so was it only acting as genetic material or anything else as well? That is also a question. Yes, it was also acting like a catalyst. I'll say it was also acting sort of like an enzyme, helping in conversion of this to this, and that was a very big trouble. Why? Because if at all you have seen the features of enzyme, they definitely change their structure altogether every now and then. That means that they do not have stability. Okay, they will not be having stability. So was that a problem? Yes, that definitely is a problem, because RNA being a catalyst was reactive and hence what? Unstable. And for a genetic material to be there, you have to be stable. Otherwise, all of a protein, whatever thing, listen, this genetic material is going to give information as to what proteins has to be made, what do we look like, and things like that. If at all that itself is changing, don't you think that everything is going to be changing every single now and then? Yes. So this was a very, very big problem. And which is exactly why, you got what? You got DNA. So from RNA, DNA was being evolved. As I said, there were changes. What were the changes? The changes were like this. In the case of an RNA, you had a ribose sugar which had an extra hydroxyl group, as in OH group at what end? At the 2' or basically the second carbon of the sugar, right? Whereas in the case of deoxyribose, it lacks one oxygen. So oxygen was being removed, which is why you got the deoxyribose. So from here you got this. Similarly, nitrogenous base, you had uracil, but uracil, my dear children, over here, from the uracil itself, what did you get? You got the new thing, which was what? Thymine. Array. What is the thing? Yes, you got what? Thymine. Now, next thing. RNA which is there, it is mainly involved in protein synthesis and transmission. So yes, we also have learned that, okay, DNA is there, but DNA transcribes into RNA and RNA gives information to a protein. But yes, we have something that is going to be there forever without much changes happening out there, right? You get it? So that is what is important. But yes, RNA also has a role. RNA is definitely going to give me information about how the proteins have to be made and also in their transport. Whereas here, the DNA is going to act as a main genetic material. Types, if at all you see, there is only one type of DNA, but in the case of RNA, you will be seeing that there are multiple types of RNA out there. mRNA, tRNA, rRNA, and the list goes on. Okay, DNA is more stable, resists degradation, whereas RNA which is there, it is less stable and degrades really, really quick. Which is why now we will go on to the next slide. And that is the criteria of a genetic material. Let's see who is the better genetic material, and let's see what is the criteria to be a genetic material. First thing, whatever genetic material is, it should be able to replicate. Why replicate? Because one cell when it is there, when it is dividing into two cells, obviously whatever genetic material is there over here has to be divided equally. That means replication. Obviously it cannot give its own DNA out there or RNA out there. It has to replicate and give the copies down. You get it? So replication, capable of self-duplication should be there, so that the genetic continuity is being maintained. Second thing, stability, chemically and structurally stable in maintaining the integrity of the organism. Okay. Third thing, mutation potential, as in it should be able to have some changes. I'm not saying much changes, but at least some changes should be there. If at all there are no changes, do you think evolution will be happening? No. So it should not be like too much changes should be there. It's like at least some changes should be allowed, because that's how evolution is going to take place. Now expression, it should be able to manifest traits in accordance with Mendelian inheritance. So however, Mendel said that, okay, this was the dominant, this was the recessive allele, whatever these things were there, that had to be followed. Okay, that had to be followed. If a gene codes for a protein, that protein should be able to be responsible for a particular character as well. That is a basic thing. So Mendelian inheritance or the expression of genes should also be there. Done. So this was the criteria. This is a very important PU2 question. Please remember it. Okay. Now, who is the better genetic material according to this criteria? If at all you talk about stability, definitely DNA is more stable. Why? Because it has complementary strands and also lack of reactive 2H group and presence of thymine instead of uracil. Listen, RNA can be single-stranded. DNA cannot, generally are not single-stranded in nature, which is why they are more stable. There is complementarity, they join together back. Okay. Now, mutation and evolution. RNA mutates faster due to instability, and RNA-based virus evolves also rapidly. Do we want this much fast evolution out there? No. We want evolution to happen in a very slow pace. We do not want high amount of mutation. It can be lethal for us, dangerous for us. Now, expression of genetic information. RNA directly codes for protein and DNA relies on RNA for protein synthesis. Yeah, that is true. Here RNA might have scored, okay. And now conclusion, DNA is preferred for genetic information storage due to stability, while RNA is better for transmission. So both of them are given a role. One is responsible for storage of all the information, one is responsible for transmission of this information or the transfer of information out there. Okay, so what is genetic material? Definitely the DNA, but RNA is also not a bad guy. Okay, yes. Now going ahead with the next thing, that is DNA replication. So we have studied about the structure of DNA, we studied about how it is packed, we studied about how it was basically surged, okay, what was the genetic material and everything, and now we are here with DNA replication process. Now DNA replication, my dear children, it was again told by Watson and Crick. So after the completion of replication, each DNA molecule would have one parental and one new strand. So listen to me very carefully. So before this, shall I, okay, I'll just cover this, then we'll go ahead. So listen to me. Over here, suppose I have the parental DNA. When parental cell, which is there, it is dividing into two cells, what is going to happen? I told the two strands of DNA are going to separate. Okay, so replication is going to happen. The two strands of DNA are going to separate and there's a new strand that is going to be formed. A new strand that is going to be formed. So one of the strand that we had is the parental strand, and the one that we have new is what? The non-parental strand. So can I say this is semi-conservative mode of replication? Can I say it's a semi-conservative mode of replication? The answer is yes. It is definitely semi-conservative mode of replication. Why semi-conservative? Because half of your parental DNA is being captured over here. It is being passed on and half of it is new. That is why semi-conservative mode of replication. This was again put down by Watson and Crick. Now obviously you needed proof for that, and that was given by Michelson and Stahl. Again a very famous experiment. It was Michelson and Stahl experiment, that is Matthew Michelson and Franklin Stahl experiment, and it was again done in E. coli itself. So listen to me very carefully. In E. coli, you were growing the E. coli first in an N15 medium. N15 medium, you know, nitrogen which is there is a part of your DNA, nitrogenous bases are there, right? So this nitrogen is taken up by the E. coli to make its DNA, which is why I say that the DNA strand, it looks like this. So okay, both of the strands had N15, N15. Now suppose I took the E. coli and I centrifuged it. What I basically got to understand is that they are going to settle up somewhere over here. The genetic material is going to settle up somewhere over here. Why? Because it is very, very heavy. What do you use for centrifugation? You use a medium of cesium chloride. You use a medium of cesium chloride for centrifugation. Where are you growing? You are growing this in NH4Cl. Here the N15 isotope is being used. Okay, NH4Cl is the medium where you are growing, and wherever you are centrifuging, that medium is cesium chloride medium. Okay, anyways. So basically they took up the DNA, and now when they are centrifuged, what is going to happen over here? You get a heavy band, because both this N15 isotope is a heavy isotope, which is why they are going to settle up somewhere over here. Okay, now we change the medium. Suppose we change the medium to N14. Okay, here N14, NH4Cl, no, in that N will be N14 isotope of the nitrogen. And then you are growing in for 20 minutes. In each 20 minutes, E. coli is going to undergo replication. That is how it processes, right? So what is going to happen is that it was seen that the DNA strand which is there, it is separating. Did they see it? No, I'll tell you how did they get to understand it. So the DNA strand was separating, this new strand is being formed. The new strand is not of N15. Why? Because you do not provide N15 in the medium, you are now providing N14. The new strand is going to be N14. Now when you did the centrifugation, you did not find a heavy band, and you actually found a hybrid band. In somewhere middle, you found what? A hybrid band. That means that probably a hybrid is being formed. A parental strand and a new strand is coming together. That is why probably I might have got this. But still, the experiment was not been over. You needed a bit more proof, which is why again for the next 20 minutes you grew it in what? N14 medium itself. That is why a total of 40 minutes has been mentioned. 20 plus 20 is equals to 40. That is what it has been. Okay, so it's not like extra 40 minutes. No, again just 20 minutes added up there. So from starting point 40 minutes. N14 medium again. So what do? This particular DNA strands are going to separate. one green and one red. The new strands which are going to come will be green only. Why? N14 is there, you're growing in N14 only, right? Similarly, these are also going to separate. What do you get? You get a hybrid strand and a next type of strand which is known as light strand. Did you find the heavy strand? No, the heavy strand is anyway gone. Why? Because we are not providing N15 medium out there. But the new strand is coming, and over here you get a hybrid. Still you get a hybrid. All of them could have been light only, but you still get a hybrid. Why are you getting a hybrid? Because the red one is still being passed down, and it will be passed on in every single generation, because the parental thing is going to be passed on. Half of it is going to be passed on for sure. That is why you got a hybrid strand out there. Got it? This proved the semi-conservative mode of replication. So was this the only proof that is what was being done by Mesos and Stahl? Yes, Mesos and Stahl only did this on Escherichia coli. I'm telling you, NH4Cl was the medium where it was grown, N15 and N14 isotopes were being used, and for centrifugation you had cesium chloride. But apart from it, other people also came in and tried to prove this. And who were those other people? It was Taylor and his colleagues. Now this is, right now PU board and also KCET has a thing for asking questions from very within the NCERT. And children tend to just ignore those texts. So please do not ignore it. This is very important. Taylor and his colleagues in the year 1958 used radioactive thymidine. So instead of using N15, N14 isotope, they used what? Radioactive thymidine to detect distribution of the DNA. And this was performed in Vishya faba. You also call this as faba beans. You also call them as faba beans. Okay. So it was done by Taylor and colleagues in the year 1958, same year itself. And my dear children, it was done on radioactive, using radioactive thymidine on what? Vishya faba, or you also call this as faba beans. Done? Okay. Now, since we have anyway spoke about replication, we need to know more about it. Okay, more about replication. How does things work out there? So listen to me carefully over here. Just give me a quick second also. No. Okay. So, suppose over here I am making something. This is your, okay, we'll go line by line, okay? This is your DNA strand. One is in 3' to 5' direction, and one will be in 5' to 3' direction. That is how it goes, anti-parallel, right? Now, the enzyme that is required for formation of a new DNA is known as DNA polymerase. What do you call it? DNA polymerase. And it is going to make a new strand only in one direction, and that is 5' to 3' direction. 5' to 3' direction. And in your whole NCERT, in all the processes, it will be in 5' to 3' only. Okay. So listen to me. In 5' to 3', that means that it has to, you have to form anti-parallel DNA, right? So it will be coming and binding actually at the 3' end, because that's how you will get the anti-parallel things. One is going to be in 3' and other is going to be in 5' 3'. So this is a strand that is going to be, this is a direction that is going to be used. Okay, 3' to 5' is the strand, and over here on the other side you have a new DNA being formed in 5' to 3' direction. I hope that's clear. Okay, fine. So first thing first, and I think I should be drawing it out of over here. So there are bonds, hydrogen bonds out there, and you need to break them down first. And for that, you need an enzyme which is known as DNA helicase. So DNA helicase comes and breaks down the bond between the two strands out there. Suppose the bonds have been broken. Now what happens? Basically, they are separate. Two strands are separate and they do not have any bonds. And always remember, it's not like the whole of the DNA is being, what do you call, cleaved altogether. No, at one particular time only a little region of the DNA is going to be open. Otherwise it is going to be highly unstable. You cannot control it. Okay, and this particular area where everything is happening, you will call this as replication fork. Replication fork, because this is the location where the replication is going to happen. It looks like a fork also. So these are the hands of the fork, and that's all, right? So that is how it goes. Listen, now since we have cleaved the DNA, there are chances that they can come together, which is why after that we need to add some proteins up. These proteins which are there, they are known as single-stranded binding proteins. So single strand which is there, they are binding on them. That is why single-strand binding protein. Okay. Now, when I say that a DNA which is there, will be making another DNA. You need to understand. Now I'm zooming this in, okay? So it is something like this. One is in 3' and one is in 5' direction. This goes in 5' to the dash and one goes in 3' to 5'. Listen, here I have opened. You had the single strand binding proteins and everything out there. Now, DNA polymerase cannot start off the process of polymerization by itself. It needs certain nucleotides already being there. You call this as the primer. The primer. And it is basically RNA primer that is going to be there. It is basically RNA primer, but small stretch which is there. Okay. Now, what is going to happen? This is attaching on the 3' end. Similarly, if at all you see over here, my dear children, where is the 3' end? This is here. Where is replication happening? From this side. From here only replication is going to happen, not from there, because that is not open, right? Only this particular portion is open. So what you're going to do is that the basic thing that you all need to know is the primer is going to come and attach over here. Okay. Now comes the DNA polymerase enzyme. It extends the DNA in this direction, that is from 5' to 3' direction, right? Here also does that. Here also, since this is 5' and that is 3' end, so from here to here, you're going to, you need patience to manage this board. Okay, so it goes like this. Agree? Everything is really smooth, but only up to a point. Why? Because every time the replication fork is going to open, here this particular strand is going to be replicated very smoothly. But what about this strand? Didn't understand? Listen to me carefully. Now over here, if at all, the picture, can I remove this? I hope I can remove this particular portion. So, here until now it was like this, something like this, right? Now, here I am going to open it more. I will be opening it more, which is why the strand is not going to look the same. It is going to look something different, right? This was my 3' end, this is my 5' end. Here I added a primer and the primer was extended. It is properly going up because anyway this is opening in the same direction. But here, my dear children, you had added a primer and from here to here you had made a DNA, because here the 5' direction goes like this. Now when you open this, again what you need is that you will again need a primer. Okay, you will again need a primer. Again the DNA polymerase will come and attach and grow it. So what do you see? You see that here the replication process is not continuous, it is discontinuous. And what do you call this as? This is discontinuous form of replication and this is continuous. This strand which undergoes continuous replication without any trouble is known as the leading strand. And the leading strand is always going to be in 3', 5' direction. Whereas this strand which does not undergo a clear form of replication, you call this as a lagging strand. And lagging strand, my dear children, is always in 5' to 3' direction. And here these fragments, okay, you get fragments of DNA out there. You call these fragments of DNA as Okazaki fragments. What do you call them? Okazaki fragments. That is what you call them. Later on, you need to fill this, you need to remove the primers. Okay, there is a process of removal of the primers and making in the DNA out there. You remove it and later you need someone to join it. Who is that someone who joins it? Ligates it? It is DNA ligase. So DNA ligase is going to join the fragments together. Wherever you see the primers, those will be removed. The DNA ligase is going to do the work. That is how it goes. So initiation, elongation, and lastly you have termination. How do you think the process of replication terminates? The process of replication is going to terminate when, my dear children, suppose from here also replication was happening, from here also replication was happening, two forks came together, okay, that's when replication can stop. Or otherwise there might be certain specific termination sites where they reach the sites, then also the termination process can easily happen. Okay, so initiation is when DNA helicase unwinds the double helix at the origin of replication. Now what is this? So replication cannot start from anywhere. There are some specific sequences which will be helping in initiating the process of replication, and this is known as origin of replication. You also call this as ORI site. Later, after DNA helicase is unwinded, single-stranded DNA binding proteins comes and attaches, and then primase adds the RNA primer over here. Later, elongation happens when DNA polymerase adds new nucleotides in 5' to 3' direction. Leading strand is made continuously, lagging strand is made in Okazaki fragments. DNA ligase will be joining these fragments later on after removing the primers. So that removal of primers is not done by this guy, but yes, joining of the fragments is done by this guy. Termination. Replication stops when two forks meet or a specific termination sites. Got it? Yes. Now, since we know about all of it, let's talk about this particular thing. Enzyme which is involved is what? DNA dependent DNA polymerase. Why you call it DNA dependent? Only if DNA strand is there, then only you can make a new strand of DNA, right? That is why you call this DNA dependent DNA polymerase. This DNA polymerase enzyme needs another strand to make a copy of it. Okay. Now, efficiency. This is a very important question. Again, NCERT, small portion, that is important. E. coli, which is having 4.6 × 10⁶ base pairs, completes replication in almost 18 minutes, I'll say 20 minutes, with a polymerization rate of 2000 bases per second. This is super important. What is the polymerization rate in E. coli? It is 2000 base pairs per second is being added. You just imagine that is how DNA polymerase is going to work. DNA polymerization occurs from 5' to 3' direction. I know that, you know that, and it requires the origin of replication. We know that. Accuracy, high precision is required to prevent any sort of mutation. Now, what is the energy source? What is going to be the energy source? So, deoxyribonucleoside triphosphate. Remember, deoxyribonucleoside as in deoxyadenosine, deoxyguanosine, deoxythymidine, deoxycytidine, whatever. So that is deoxyribonucleoside, and then you have triphosphate. That means deoxyadenosine triphosphate. What do you mean by that? I told you, whenever you are making a nucleotide, it does not look like this. 1, 2, 3, 4, 5. Over here you have the nitrogenous base. Over here you have the phosphate group. But the phosphate group does not come like this. There are 3 phosphate groups out there. So basically, this is deoxyribose, right? Deoxyribonucleoside out there, and over here these are the triphosphates. Now when these bonds are going to break, what is going to happen? They're just going to produce too much amount of energy that will be helping in the formation of another bond with another nucleotide. So always remember, this guy is the substrate that you need. It is what? Something that you need to make the DNA. That is what you call as substrate. And also this is going to be the energy source. Why energy source? Because, my dear children, when this bond is broken, high amount of energy is being released. So it is an energy source. What is energy source? Deoxyribonucleoside. Okay, that means nitrogen is also there. Triphosphate. So you call this as DNTPs. This is the basic structural unit of DNA. Always remember, don't just go write nucleotide and come. It is deoxyribonucleoside triphosphates. Fine. So yes, these are the substrate as well as energy providers. That's all. That ends up right over there. And now we go ahead with the next thing, that is how does DNA help in the formation of proteins? So how does DNA help in the formation of proteins, my dear children? I told you about one very important thing. The DNA which is there, it forms the RNA, and RNA which is there, it forms the proteins, or it helps in the formation of proteins. It gives the information. This process is known as transcription. This process is known as translation. Okay, when you make DNA out of DNA, you call this as replication. This is the central dogma. This is how generally things work. That is why we call this as central dogma. This was given by Francis Crick. This was given by Francis Crick. And now I'll tell you how things are going to take place. Your DNA definitely cannot move out of the nucleus, because this is something that we need. This is a major source. So DNA lives inside the kingdom itself. The DNA undergoes transcription to form the RNA. the mRNA, and the mRNA is something that is going to be transported outside. Why outside? Because I said RNA needs to form the proteins, and for forming proteins it needs some very important things. For example, you might have heard about ribosomes. Yes, so ribosomes are there in the cytoplasm. Similarly, it needs tRNA, different type of RNA that is also there. So all of this is there in the cytoplasm, which is why it goes out for the process of translation to happen. In the case of eukaryotes, this is like this. In the case of prokaryotes, do they have any nucleus and things like that? No. So everything happens simultaneously itself. Transcription, translation, everything happens simultaneously. But for us, it is highly regulated. Okay. So from this point onwards, we have to be fast because I see the timing is a bit more. Yes. Let's go ahead. So what does the transcription unit contain? So this structure that you see over here is a transcription unit. You basically need these things for transcription to happen smoothly. What is this? Let me see. So one is the DNA strand, which is in 3' to 5' end. Other one is basically another strand which is in 5' to 3' end, right? Now, here also RNA polymerase binds at the 3' end and makes the new RNA in 5' to 3' direction. Got it? Which is why I'll call this strand which is there as template strand. On the other hand, I'll call this strand as coding strand. Coding strand does not undergo transcription, it's just there. It does not undergo transcription. This is the guy that undergoes transcription. Only the template strand. Now, we say that in a transcription unit, you need a specific region, that is promoter region. Onto this promoter region, the RNA polymerase will come and bind. But where is it located? You say the location of promoter is in the 5' end of the coding strand. You give little bit work to the coding strand, right? So that is why you're saying in the 5' end of the coding strand, you have the promoter region. Similarly, at the 3' end of the coding strand, you have the terminator region. So you have promoter region, you have terminator region. Onto the promoter region, the RNA polymerase will come and attach. On reaching the terminator region, RNA polymerase will stop the process of transcription also. Got it? So transcription unit of DNA is defined primarily by 3 regions: promoter, okay, structural gene, so this is the thing that is going to be coded, and then you have the terminator. Transcription is done by DNA dependent RNA polymerase. Again, do you need a DNA? Yes. So which is why you say that it is done by DNA dependent RNA polymerase in 5' to 3' direction. Okay, additional regulatory sequences may be present further upstream or downstream of the promoter. This is also given in your NCERT, that it can be some additional regulatory sequence can be present over here and over there also in some organisms up there. Okay, now how does the transcription works? This is the easiest among all of them. So listen. The RNA polymerase says that I cannot go and bind to the promoter region alone. I am shy. So since it is shy, it needs a factor, and that factor is known as what? Sigma factor, my dear children. So shy stands with S and starts with S. Here sigma factor also starts with S. So initiation is when RNA polymerase binds to the promoter with the help of a sigma factor. The DNA helix opens and the transcription is going to begin. So for binding to the promoter, you need the sigma factor. Again, a very important thing. Next thing, elongation. RNA polymerase add nucleoside triphosphate NTPs, okay, not deoxynucleotides, nucleoside. Remember, it's just nucleoside over here. It is not deoxy, it is just nucleoside triphosphates. Why? Because here you are forming RNA, not DNA. So RNA polymerase adds nucleoside triphosphates. Following the complementary base pairing rule, only a short stretch of RNA remains attached to the enzymes. So it goes on, and only a short stretch is going to be attached, rest is going to be hanging like this. Okay, and the strand is just getting formed. So if at all you see the structure, it looks like this. It is going. By the way, sigma factor went off. Okay, after initiation, sigma factor went off, and now this guy is going alone. Now, my dear children, when it reaches the terminator sequence, again it needs a factor to terminate. So it is crying that, oh my god, I have to leave the DNA, it's my friend. So it is crying. You call crying as Rona in Hindi, and for rho you have rho factor. Okay, so rho factor comes and consoles the RNA polymerase and say that, bye, come, let's go. Mucha we have to go. So rho factor is there for what? The termination process. RNA polymerase reaches the terminator region. Rho factor helps in termination, causing the RNA to fall off. RNA polymerase detaches, completing the whole transcription as done. Done. That's all. So initiation, elongation, termination. For termination you need rho factor. For initiation you need sigma factor. Now you can remember like that. Initiation, there is a s sound over here, so you need sigma. Termination has R sound more, so it is rho factor. You can remember like that also if at all you want. Now over here you have two things. One is prokaryotic transcription, another one is eukaryotic transcription. What do you mean by that? What is the difference? Just a second, okay? So listen to me. Over here, we say in the case of prokaryotes, my dear children, they have, for example, if I say that transcription is happening, they will be having multiple genes, and these multiple genes will be coding for multiple proteins. You call this as a cistron. And I'll say basically they are polycistronic in nature. They will be polycistronic in nature. They have multiple cistrons, okay, and multiple cistrons, basically multiple genes and multiple proteins are being formed. So they are polycistronic, the multiple genes are there that codes for multiple proteins. But in our case, we are monocistronic. That means we just have, in a cistron, we just have one gene, and that one gene is going to code for one particular protein. One particular protein, that's all. Okay, so that is eukaryote and prokaryotic transcription difference. They are polycistronic, they have multiple genes coding for multiple proteins. We are monocistronic, one gene codes for just one protein. Example might be asked, you can ask pea plant, what is it, polycistronic, what do you call, polycistronic or monocistronic? It is monocistronic. Why? Because pea plant is what? A eukaryote. So please know about that. Next thing, and that is prokaryotes do not have introns. What do you mean by that? So basically in our DNA, there are regions that codes for proteins. You call these regions as exons. And there are regions that do not code for any proteins. You call this region as what? Introns. Introns, the non-coding ones, right? So whatever information is there in the DNA that is being transferred to the RNA, and that RNA, it's not mRNA, it is actually hnRNA, heterogeneous nuclear RNA. So here also you will be having exons and introns. Exons and introns. But do we need these introns? No. While we go ahead with the formation of proteins, we do not need them because they are not going to code for any protein. So basically, this hnRNA that you have undergoes some processing and makes another type of RNA that is known as mature RNA or messenger RNA. In this RNA, you will remove all the introns and you will be just having the exons. Only the coding sequence. Okay, just understand that. So if at all you are asked what is common between hnRNA and mRNA, it is just exons. Introns are anyway removed. Now, in the case of prokaryotes, they do not have introns. So do they need this processing and things like that? No. Okay, whereas in the case of eukaryotes, we definitely have this. We definitely have introns, and that has to be removed during the RNA processing. And as I told you, transcription and translation occurs in the same area because they do not have any nucleus, this and that. So everything happens in the same area, that is cytosol. Whereas in the case of eukaryotes, transcription and translation occurs at different location. Transcription at nucleus and translation in the cytosol, or basically what you call as cytoplasm. So here they are polycistronic. You can remember like this, P and P. Prokaryotes are polycistronic. They have multiple genes, multiple proteins being formed. Okay, and so that means basically one structural gene which is there. Here multiple proteins can be made. They have multiple cistrons, okay, then they code for protein. So over here, us in our case, one structural gene, that means only one gene is being considered, no multiple genes. Okay, you will get an understanding of it when we study about lac operon. Okay, so that is how it goes. I hope this is clear. Prokaryotes do not have introns, we have introns, and yeah. Now, RNA polymerase and RNA processing in eukaryotes. Let's talk about that. So we have three types of RNA polymerase. In the case of prokaryotes, they just have one, but we have three types of RNA polymerase, and all of them do different things. Let's see what. So RNA polymerase 1, the first priority, 1 means the first priority, you can think like that, is to form rRNA. rRNA means your ribosome, which is the organelle, no, it is made up of ribosomal RNA. So this rRNA is that one. Okay, and in rRNA of different types, you have 28S, you have 18S, you have 5.8S. So you have 28S, 18S and 5.8S. They are all RNA polymerase which transcribes the rRNA. Okay, got it? Sorry, did I mean something wrong? Just a second. Okay, so RNA polymerase 1 is responsible for transcription of rRNA, and there are different types. That's all. Forget about everything. And all of them ends with 8. 28S, 18S and 5.8S. Now going ahead, RNA polymerase 2. Second function that we have is to transcribe hnRNA. It is a precursor of mRNA. So once you have hnRNA, then only your mRNA can be formed. So this is a second priority. Now third priority, third and T is for tRNA. The next type of RNA that is there is transfer RNA, and that is being transcribed with the help of RNA polymerase 3. Apart from that, some type of rRNA which you also call at 5S rRNA and some small nuclear RNAs are also being transcribed from this. So remember, it is tRNA, 5S, okay, rRNA. Since everything was ending with 888, it was not included there, so it is included over here. 5S rRNA and also small nuclear RNAs. Small nuclear RNAs are also being transcribed by RNA polymerase 3. Okay. So RNA polymerase 1 rRNA, polymerase 2 hnRNA, heterogeneous nuclear RNA, which is the precursor of the mRNA, and last one is this one. Now RNA processing, as I told you, RNA which is being formed, this is hnRNA. You have to process it and make it turn into what? mRNA. And for that there are certain things. One is splicing. Splicing is where you remove the introns and join the exon. Done. Splicing is done. Fine. Next is capping. Capping means basically you add methyl guanosine triphosphate at the 5' end. At the 5' end of this thing, what are you going to add? Methyl. It looks like this, okay? Methyl guanosine triphosphate. Methyl guanosine triphosphate. You are adding up over here in the 5' end. And in the 3' end, that is known as capping. In the 3' end, you add multiple AAA adenine residues. So you call this as polyadenylation. Why are you doing this tailing process and capping process? Such that as of now, until now, it was there inside the nucleus. You know, mothers, when they send you outside, they put sweater and things like that. Similarly, our nucleus also is sending it outside with due protection, because once mRNA reaches outside, there are chances that it can get cleaved. So that is why you have the capping and the tailing process being done. So capping means methyl guanosine triphosphate at the 5' end, and tailing means almost 200 to 300. Almost 200 to 300 adenylate residues at the 3' end. Fully processed mRNA is transported to the cytoplasm for the process of translation, for the next process, that is translation. This is how things work. Done? Done. Okay, fine. Now, next thing that we come across is the genetic code. The genetic code. So that's only how you can understand translation process. So when I say an mRNA is being formed, it has the information to form the proteins. But how do you think that is being formed? So you have sequences. For example, you have A, U, G, and you have C, G, A, and things like that. Always remember, it's RNA, it will be having U instead of thymine. This is a strand that you have. How do you think these strands which are there can give rise to information to form a protein? Because, my dear children, actually these are codes. These are some secret codes out there. Each code which is there, it is going to code for an amino acid. And each amino acid which is going to be formed together will be forming what? The protein. Together will be forming what? The protein. So listen, who told that the genetic code which is there is going to be triplet in nature, going to be 3 in number? It was George Gamow, a physicist who told that it is going to be triplet, because according to him, we have how many nucleotides, my dear? How many nitrogenous bases? We have A, U, G, and C. Suppose we have 4 of them, right? And 4 of them, if they come in the pair of 2, so we get basically 16. 16 proteins can be made. 4 of them, if they come in two pairs, 16 codes will be there. 16 codes can maximally code for 16 proteins, right? But how many proteins do we usually have? Proteins, amino acids. So 16 amino acids can be formed, right? But how many amino acids do we generally have? We have 20 amino acids. So this is not possible. We have to make 20 amino acids. 4 of them are still less, okay? So what do we do? Okay, then probably the 4 of them, if they come in this format, then probably it will be 64 of them. So ma'am, we just needed to make 20 of them. But what could we do? We do not do anything in between, right? Obviously. So there is only one option. Have extra codons. That is totally fine. Have extra codons. Some codons can code for the same amino acid. For example, UCU, UCC, UCA, UCG. Everything codes for the same amino acid as a serine only. So repetition can be possible. But anyway, 20 amino acids can be coded with it, right? Yes. So this was the idea of George Gamow, who said that yes, it comes in triplet format. Now, what are the features of genetic code? This is a very famous question that is usually being asked. So the codon is going to be triplet. This is the feature, first feature, the codon is going to be triplet. How many codons do we have? 64 codons. Out of the 64 codons, 61 of them are going to code for amino acids, and 3 of the codons do not code for any amino acids. 3 of them are not going to code for any amino acids. They are going to, if at all they are there is nothing going to be coded. It will stop the process of translation, the next process that is going to happen. What are these codons? I'll be talking about that here itself. So UAA, UAG, UGA are the stop or the terminator codons. What is this? UGA, UA and UAG. They are what? They are the terminator codons that you need to remember. Okay, and they are triplet. 61 of them they code, but 3 of them that are this, they do not code for anything. Okay. Next thing, some amino acids are coded by more than one codon, hence the code is degenerate. What do you call this feature as? Now they can give you a scenario where they are saying that basically serine which is there is coded by this. What feature of genetic code is being seen over here? This feature is known as degenerate feature, where multiple codons are coding for the same amino acid. So I call this as degenerate feature. And always remember, a codon which is there will code for only one amino acid. A codon at a time will code for only one amino acid. This is known as unambiguous nature. Do you see UCU anywhere over here? No, you will not be able to find. So one codon will code for only one amino acid, that is unambiguous nature. And when I say multiple codons are there for a single amino acid, I'll say this feature as degenerate feature. 2 of things are different. Okay, so basically UCU thinks that I am the only one coding for serine. But then it is UCC also, it is UCA also. So this is degenerate feature, no? So you can remember like that. Okay, works? So yes. Next, the codon is read in mRNA in a contiguous fashion. So mRNA, in the codons which are there, they are read without any full stop, any commas out there. So that has been mentioned over here. The code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine. Yes. So for many organisms that you see, the codons are going to be the same. They are going to code for the same amino acid. There are exceptions, and you need to know about exceptions because you get questions. So basically, exceptions are what? Exceptions are found in mitochondrial codons. In mitochondria also you have DNA, you have RNA, they also have codons. So in that codons, their things can be different, and also some protozoans. Okay, some protozoans in there also it can be different. So mitochondrial codons and also some protozoal codons, their things can be a bit different. Rest everything is same. So I have told 61 codons code, 3 codons do not code. Some amino acids are coded by more than one codons, that is known as degenerate feature. They are read in contiguous fashion. It is nearly universal. There are 3 stop codons: UAG, UAG, UGA, UGA, and UAA. Okay, now there is the first codon which is there, that is known as AUG codon. And AUG codon has dual function. One is that it initiates the process of translation. So every time the first codon is going to be methionine itself. Sorry, first amino acid is going to be methionine itself. Why? Because AUG is the codon coding for methionine. And not just that, it also, as I said, it has dual function. One is to code for methionine, and the second one is to act like a initiator codon. So just like how we have terminator codon, we have initiator codon. Now, listen to me carefully. So generally it is being said that you need to remember the codons for KCET, not for PU2, for KCET. But majorly, if at all I talk, there are a few things that you need to keep in mind. Methionine is a codon that is coded by only one amino acid, that as I told you, it is AUG. Similarly, there is tryptophan. And tryptophan is also an amino acid that is coded by only one amino acid, one codon, which is UGG. UGG. Okay, 3 stop codons name you know, and UUU is generally asked, it is phenylalanine. UUU is phenylalanine. UGG is what? Tryptophan. UGG tryptophan, AUG methionine, both of them are coded by only one codon. Cool? Yes. Next. So, scientists behind the findings of all this thing, how did they came up to this conclusion? So, first thing, there was a person whose name was Severo Ochoa. Okay, he came up with an enzyme that is polynucleotide phosphorylase. What was this enzyme? This enzyme can make RNA without the help of DNA. Without the help of DNA, an RNA could be made. What was the enzyme? It was polynucleotide phosphorylase. Okay, polynucleotide phosphorylase. That was the name of the enzyme. It did not need DNA to make the RNA. With the information taken by from Severo Ochoa, next person, that is Hargobind Khorana, came into the picture. Hargobind Khorana, actually, you got the enzyme, right? You got the enzyme. So why not synthesize a whole RNA? So he was a person who chemically combined RNA sequences, codons out there, and made an RNA. So chemically synthesized RNA sequences to test how codons actually work. And now, on the basis of this, Marshall Nirenberg came into the picture. And Marshall Nirenberg, my dear children, developed another self-free system to experimentally confirm which codons code for which amino acid. So until now, everything happened inside the cell, but now he created a cell-free environment where he could identify that, okay, this codons which are there, they actually code for these amino acids. And with that, we were able to identify the codons as well as their amino acid. So Severo Ochoa gave me the enzyme for formation the RNA, Hargobind Khorana made the RNA, and then Marshall Nirenberg came into the picture and basically created a cell-free condition where translation can happen, and he could understand that what sequences of RNA, because while this Hargobind Khorana made the RNA, he knew what was the codons out there. He had a real idea about this. Now there should be someone to make the amino acids, right, and that was Nirenberg. So people got to understand that, okay, this amino acids are being formed, so probably it is coded by these codons out there. That is the logic. 3 things. Remember. Done. Okay. Next thing. So here I have a chart so that you can remember things. I'll give you a hint and then I'll go ahead. So fill leaf in my wallet. So fill leaf in my wallet. This is how you can remember. Fill as in phenylalanine, leaf as in leucine, in as in isoleucine, my as in methionine, and valid as in valine. So this is a famous thing out there. If at all you want, you can make the best use of this particular thing also. Okay, similarly I can just tell you. So serious professor through alarm, serine, proline, threonine, and alanine. Serious professor through alarm. Then you have what? Tyrosine, stop his gun. So tyrosine, stop his gun. So tyrosine and stop codon, histidine, and you have glutamine. Apart from that, Asian like aspartic glue. So Asian like aspartic glue, that is asparagine, lysine, aspartate, and you have glutamate. Fine. And then last you have sister to stop tripping. So sister, stop tripping. Okay, that is cysteine, stop, and tryptophan. And over here, what do you have? You have arginine, arginine, glycine. That does not make any sense, but yes. Arginine, serine, arginine, glycine. Okay, serine is repeated twice here and here, so be sure you check it up. Yes. Now, we go ahead with the next thing, that is mutation. So insertion or deletion of any or 1 or 2 bases change the reading frame from the point of insertion or deletion. This is known as frame shift mutation. So listen to me carefully. You had Ram has red cap. If at all you're inserting B over here, what is going to happen? Ram has BRE. So over here the reading frame has changed. That means the amino acid which is going to be formed is also going to be changed. So basically reading frames are going to be changed if at all you insert one nucleotide or one more nucleotide. So 1 or 2 nucleotides if at all they are inserted, or 1 or 2 nucleotides if at all they are deleted, it can lead to what? Frame shift mutation. The frame of the reading is going to be shifted, because anyway reading is going to happen only in triplet codons. You got it? If at all you are inserting 3 of them, it is not going to shift any frames, but you need to understand, suppose I am inserting 3 codons, that is UAA. They are stop codons. It will stop the process of translation. It can stop the process of translation. So just have that idea. Cool. So 3 codons, usually they do not change any frames, but they can stop the process of translation if at all they are the stop codons. Okay. Now, We'll go to tRNA and then we'll talk about translation process, and then we'll talk like operon, and slowly we'll end up. Okay, so tRNA, which is there, is known as soluble RNA also, or sRNA also. What does it do and how does it look? So my dear children, it is just a normal RNA which is twisted and turned like this. Hi, why is it happening like this? Just a second. Somehow I'm not able to make it. Just a second. Okay. So this is your 5' end and this is going to be your 3' end. Okay, this is an RNA. Now over here you have your mRNA. mRNA in what direction? 5' to 3' direction. Over here you have codon, suppose you have AUG. So what exactly happens is that this tRNA which is there, at the 3' end, it is going to be attached to an amino acid. This process is known as amino acylation, and also understand this is also known as amino acylation. Apart from that, it is also known as charging of tRNA. Charging of tRNA or amino acylation. Now, whenever you do that, you need high amount of ATP to form this bond. High amount of ATP to form this bond. And also, every time a tRNA is attached to amino acid, just understand it will also have an anticodon over here. An anticodon loop over here. So here it was AUG in the codon, right? The anticodon of this will be A base pairs with U, U base pairs with A, and G base with C. So over here in the anticodon loop, you will be having an anticodon opposite codon off to this, and you will be having an amino acid coded from this. You get it? That is how things are going to work. So tRNA is also known as soluble RNA or sRNA. Okay, S, not snRNA, that is small nuclear RNA. This is sRNA. So there is an amino acid acceptor end where the amino acid comes and attaches. You call this process as amino acylation or charging of tRNA, and then there is an anticodon loop right over here. Okay, the secondary structure of tRNA has been depicted that looks like a clover leaf. Secondary structure of every protein or every RNA that you say, you have primary structure, secondary structure. In the case of secondary structure, you have a clover leaf structure. In the actual structure, tRNA is compact molecule which looks like inverted L. It looks like what? Inverted L. Cool, it looks like inverted L. So secondary structure looks like clover leaf, but the actual structure is what? Inverted L. Please remember that also. Fine. So we had the tRNA. You got an understanding of what exactly is tRNA. Now listen to me carefully. What is going to happen? So it goes like this. First thing that happens is amino acylation. As I told you, it is super ready, right? Amino acylation of what? Of tRNA or charging of tRNA. That is happening. So tRNA are all charged. They have the amino acids ready. You have the mRNA now. Onto this mRNA, what is going to come and bind? The small subunit of ribosome. The small subunit of ribosome is going to come and bind. Now the third thing that is going to happen is that this mRNA plus the small subunit, it has the initiative codon AUG. Onto this, you will be having the tRNA coming and attaching with methionine. Methionine is the first amino acid being formed, right? And not just that. After this, you have the larger subunit also coming and attaching. So initially small subunit came and attached, then you had the tRNA and also the large subunit. Together now we'll call this complex as initiation complex. Now the process is going to begin. This is how it goes. So basically the ribosome which is there, it is going to read, it is going to move in this direction. What direction? Again 5:3 direction. So whenever it moves, a tRNA will be coming and attaching to those areas. Okay, when the tRNA is coming and attaching, for example I say here it is attaching with some another amino acid. What is going to happen? These two amino acids which are there, they are going to form the bond, and this tRNA which is there, it is going to move out. What is this bond that is going to be formed? This is the peptide bond. Between the amino acids, what do you have? You have peptide bond. Similarly, you will get a chain of amino acids like this, which is finally known as what? The protein. That is what usually happens. So over here, you have enzymes, RNA, which is made up of rRNA, I told you, ribosome, which is made up of rRNA. rRNA has some catalytic properties, just a second. I'm getting confused. Okay, yeah. So, yeah, I'm making you confused. That is what I meant. So, ribosome which is there, it has rRNA and some very important proteins. Ribosomal RNA is rRNA and some proteins are also there. These proteins which are there, they are going to have some catalytic functions helping in what the bond formation as well. Similarly, rRNA which is also there, that also has some catalytic functions. Cool. Now, I will not make it complicated, so it is little higher level things. So, let's read it out and yeah. So, activation or charging of tRNA is the first thing that happens. Okay, initiation, mRNA binds to a small ribosomal subunit. The start codon is recognized by initiated tRNA, and then as I said, the larger ribosomal subunit also assembles, making the initiation complex. Now, elongation, tRNA brings amino acids to the ribosomes, matching codons on the mRNA through its anticodon. So, tRNA comes on the binds on the codons through the anticodon loop. Now, peptide bond forms between the adjacent amino acid catalyzed by a peptidyl transferase. That's an enzyme. Peptidyl transferase. Now, ribosome also acts like a catalyst. Why? As I told you, there's an enzyme over here. We are talking about the enzyme, that is peptidyl transferase. That is basically a protein that forms the ribosomes. A protein is there in the ribosome that is doing the function. Apart from that, it is said that the ribosome as a whole also acts like a catalyst. Why? Because ribosome is made up of rRNA and the protein, right? And this type of RNA, 23S rRNA, in bacteria is the enzyme ribozyme. Is actually the enzyme ribozyme that makes up the bonds out there. So yes, there is peptidyl transferase that is separate. In the case of bacteria, there is a type of rRNA that is known as what? 23S rRNA, and that acts like a catalyst as an enzyme, and this is known as ribozyme. Also remember that. Done. Okay. Termination. So when a stop codon is reached, obviously there is no tRNA that is going to attach on the stop codon. Instead of that, a release factor can come and attach on the stop codon out there, but never a tRNA. Release factors can come and attach, but never a tRNA. Now there is a very important thing that is usually being asked, and that is untranslated regions. So listen to me carefully. When I said that the mRNA is getting translated, the mRNA, this is a 5' end, this is a 3' end. Before the 5' end, there will be some region. After the 3' end also, there will be, okay, if I go there, it will be problem. Okay, after the 3' end also, there are certain regions. What do you call these regions as? You call these regions as untranslated regions. What do you call them? You call them as untranslated regions, or you also call them UTR. What is the use of that? UTR's use is that it makes the mRNA stable. Second thing, it allows the process of initiation to happen. It allows the process of termination to happen very smoothly. So if at all you want translation to happen, you need UTR, because UTR maintains the stability of mRNA, and for the smooth initiation and termination, you need UTR. Remember, it contains untranslated regions at the 5' end and 3' end. That means before, not after the 5' end, okay? Before the 5' end and after the 3' end. You say upstream of 5' end, that means before it, and downstream of 3' end, that is after it. Okay? Got it? Yes. You have to be very careful. They can, clearly make you confused as well. Fine? Yes. Now, since we have studied about all of this thing, now we need to talk about lac operon. So lac operon, my dear children, you got an understanding that in the case of prokaryotes, things are very different from eukaryotes, right? And I told you, we learned that apart from promoter region, there can be some other regions also, upstream or downstream, that can be doing the regulatory function. Remember when we spoke about the term transcription unit, we said that upstream or downstream of the promoter, you might have some regulatory sequences, right? So lac operon is one example for this. Lac operon is seen in E. coli, and basically E. coli will be needing either glucose or lactose for energy source. Suppose it is grown in a medium where there is no lactose, things are going to be different. If there is lactose, things are going to be different. So I'll give you two scenarios, and it goes like this. So over here, there is a sequence. Okay, there is promoter region, and this is the gene. You call this gene as regulatory gene. What do you call this? Regulatory gene. It will be coding for a protein that is having negative regulation. This protein is known as repressor protein. So regulatory gene over here is doing negative activity. It is producing what? Repressor protein. Okay, so there is another promoter region and another set of gene over here. For this set of genes, you have a separate promoter region, and not just that, you will be having an operator region also. There will be an operator region, right? And these are the genes Z, Y, A. I told you they are polycystonic, multiple genes out there, right? So Z actually codes for beta-galactosidase, Y codes for permease, and A codes for transacetylase. Beta-galactosidase is the one that is going to break down lactose into glucose and galactose, providing energy. Permease is one that increases the permeability of the membrane such that anything can come inside, especially lactose. Transacetylase deals with anything in between. But as of now, when there is no lactose, do you think all of them need to be produced? No. So transcription of them, do you think that should happen now? No. Which is why the repressor protein which is there goes and binds onto the operator region. Why? Because RNA polymerase will come and bind on the promoter region and will try to do transcription, but it will not allow. Why? Because something big is coming, sitting in the front, and that is onto the operator region. And what is that? That is a repressor protein. So in this condition, none of the genes are going to be transcribed. So I'll call this condition as switched off condition. What do I call this? I call this as switched off condition. On the other hand, suppose the E. coli is grown in a medium that has lactose. What is going to happen? The lactose enters the cell. Okay, the lactose will enter the cell, and then what is going to happen? The lactose which is there, it says that repressor, bro, don't go and attach on the operator. Basically, I think E. coli needs me. You be here. So in it, the lactose which is there, it binds to the repressor and does not allow it to go and bind on the operator region. And what is going to happen? Transcription is going to happen. Now you have beta-galactosidase, you have permease and transacetylase, all of them being formed. So in this condition, it is making the lac operon switched on. The one that is doing this is known as inducer. And what is that? It is lactose over here. So it can be either lactose or it can be also allolactose, a different form of lactose. So it can be lactose or allolactose that acts as what? That acts as an inducer and goes and binds up there. Got it? I hope you got it. It's very, very, very important thing. Okay, now they will do that. So now beta-galactosidase is there. The lactose which is there in the medium is being taken up. It is being broken down into glucose and galactose. Permease increases the permeability such that more lactose can come inside. Okay, transacetylase, as I said, deals with everything in between. So ma'am, how did lactose come inside if at all permease was not being transcribed? So it is not like that. Even in switched off condition, little bit of transcription definitely happens, which is why it was permeable enough to get inside. Okay? Yes. So please know the sequence of it. You have the promoter region for the regulatory gene, that is I gene. Then you have promoter, then you have operator, and then ZYA, ZYA. You can remember like that. This is very, very important. And what is the role of lactose or allolactose? They are what? Inducers. They induce the process. Done. Now, we come into the next thing, and that is what? The Human Genome Project. By the way, before I go into that, in what all levels do you think that the transcription or gene expression is regulated in the case of eukaryotes? In what all levels do you think gene expression is going to be regulated in the case of eukaryotes? So gene expression means from the DNA to protein. How can it be regulated? So at first level is transcription level. I can stop this process to happen. Second thing is processing level. I can stop the processing of hnRNA to mRNA. Right. Third is transport of mRNA. Fourth is translation of mRNA. At these four levels, I can regulate the gene expression in the case of eukaryotes. Very important question. At four levels: one, transcription level; processing level; third, transport level; and fourth is translation level. In the case of prokaryotes, everything happens in the same region, so we cannot regulate it much. Just like how you have the lac operon, you have regulatory genes regulating a lot of things out there. Okay? Yes. Ayo. Now we go to Human Genome Project. Where is it? Yes, we are here. And we are going to soon end the class, don't worry. So what was the aim of Human Genome Project? To identify approximately 20,000 to 25,000 genes in the human DNA, determine the sequence of 3 billion chemical pairs that makes up the DNA. Okay, 3 billion chemical pairs that makes up the DNA. To store, okay, so first is to approximately identify the genes, how much? 20 to 25,000. Second is to what? Basically identify the sequences of each gene, that comes out to be almost 3 billion chemical bases, okay, and then store the information in some database, and after that provide some tools or improve the tools for data analysis, and after that transfer it to the related technology areas where things are being developing, and the last one to address the ethical, legal and the social issues that may arise from the project. So initially PU board used to give direct questions like this, what is the aim and things like that, but now and all they are twisting things, okay, they are asking deep inside, so please make sure you know about it. So, Human Genome Project was a 13-year-old project coordinated by the US Department of Energy and the National Institute of Health. Who all? It is US Department of Energy and also the National Institute of Health that came together to give the Human Genome Project. It was a 13-year-old project out there. What is it? US Department of Energy and National Institute of Health, NIH. Please know about that. Now, in the preparatory examination that I saw a question like this. Okay, so obviously many human beings genome is being studied and things like that is happening, but are there some non-human model organisms as well? The answer is yes, and you need to remember their names, such as bacteria. It can be yeast, there can be Caenorhabditis elegans, that is a free living non-pathogenic nematode. There can be Drosophila, there are plants such as rice and Arabidopsis that also have been sequenced. What is it? Bacteria, yeast and C. elegans out there. And what do you have? You have Drosophila, and you had what do you call plants like rice and Arabidopsis also been sequenced. Please know that they are also model organisms. Cool. Next thing. What did you find in the Human Genome Project? What did you find in Human Genomic Project? So Human Genome contains 3,164.7 million base pairs. Remember, each and every data over here in numbers is in millions, not in billions. So 3,164.7 million base pairs were there. Okay, in the whole human genome, and the average gene contains around 3,000 bases. How much? 3,000. Average gene is having 3,000 bases, but there is an exception. There is dystrophin, which is having 2.4 million bases. 2.4 million bases. Dystrophin is a protein. Okay, so the average size is what? The human genome size is what? Not average size. The human genome size is 3,164.7 million base pairs, and the average gene size is average gene size is 300 base pairs. Okay, only 300 base pairs will be there in average genes out there. But now when you talk about dystrophin, they have 2.4 million base pairs. Now they can ask you a question like this: HNRNA. What is the average gene size of HNRNA? It's the same thing, only it's 3,000 bases, because DNA is exactly put down into HNRNA, right? So whatever thing will be there will be here also. So 3,000 base pairs. Okay. Now the total number of genes estimated is 30,000, much lower than the previous estimates of 80,000 to 1,40,000. Okay, the functions are unknown for over 50% of the discovered genes. Even though we have discovered it, but 50% of the discovered genes we do not know their function. Less than 2% of the genome actually codes for proteins. Only less than 2%. Less than, not more than, not equal to. It is less than 2% of the genome that actually codes for what? Protein. Repeated sequences makes a very large portion of the human genome as well. So rest everything. So when I say only 50%, okay, so I am saying that only less than 2% of gene actually codes for proteins. That means rest of it is what? Rest of it would be making the repetitive sequence. These repetitive sequences, they vary from one organism to another, and with the help of understanding of these repetitive sequences, how do they differ between one organism to another, we can understand how we have evolved as well. So they have a role in understanding the evolution also. Now, chromosome number 1 has the most number of genes. What are the most number of genes? It is 2968. It is what? 2968. Chromosome number Y has the fewest. It is what? 231. Remember, this is important. Okay, so chromosome number 1 was the first one. Okay, you need to know that also. Chromosome number 1 was the first one to be identified fully, and chromosome number Y, and also it had the highest number of genes, that is 2968. So only the number 7 is missing, rest every number is there, right? And chromosome number Y has the fewest, that is 231. Now, scientists have identified about 1.4 million locations where single DNA difference is there. Okay, you and me differ only at one nucleotide position, that is known as single nucleotide polymorphism. SNPs, single nucleotide polymorphism, that is also found at 1.4 million locations like that. That's how we can be different. Okay, so please remember all this information, the one that I have marked in yellow are super important for you to know. Okay, works. Now, there was two ways of sequencing the human genome. One was expressed sequence tags, ESTs. Expressed sequence tags means you only code, basically decode the genes which are coding for things. That is only exons, not the introns. That is express sequence tags. Second one is whole genome sequencing. The whole genome entirely, whether it's coding, non-coding, everything was sequenced. So please remember that also. And that was done by sequence annotation method. Okay, that was done by, so whole genome sequencing was done by sequence annotation method. And Frederick Sanger, okay, there was a person Sanger who had a really big role in sequencing. Sanger's method was being utilized over here and he had a really big role in sequencing. Cool? Done? Done. Okay, next thing. What do we have? DNA fingerprinting, the last topic for today. So human genetic variations. So over here, the human DNA is made up of 3 billion base pairs. Agreed? Agreed? 3 billion base pairs? Okay, agreed. We studied right now, right? 3,164.7 billion base pairs. Sorry, million base pairs. So when you say 3,164.7 million base pairs, you mean 3 billion base pairs. Okay. 99.9 of this DNA is identical in all humans, meaning only 0.1 differs. You and me have 99% of DNA as a common thing. 0.1 difference is there. But 0.1 of 3 billion is equals to what? 3 million base pairs. Okay, is what? 3 million base pairs. So we are going to, whenever we say only 0.1 difference is there, we still mean the 3 million base pairs in you and me are going to be different. This means that 3 million base varies between individuals, making person unique in their traits. Now, DNA fingerprinting overview. Instead of sequencing all 3 billion base pairs to compare two people, scientists focus on specific DNA regions that are different. These variable regions are known as repetitive DNA, contain small DNA sequences that repeat multiple times and vary between individuals. So, my dear children, we have two types of DNA. One is the bulk DNA which is there, which actually codes for the protein and things like that, and the other one is satellite DNA. The satellite DNA is something that is made up of repeated sequences and they are not going to code for anything. So we are going to see the differences happening up over here. Okay, we are going to compare between organisms on the basis of this, because we might be sharing some repetitive sequences. For example, I might say, I'll tell you one example. Okay, there are type of repetitive sequence which you call as variable number of tandem repeats. You know what's very unique about them? You and me have the repeats. For example, I say it is AGCT. You and me have it. I have it 14 times. You have it probably 8 times. So repetitive sequence are there, but the number of the time it is getting repeated is also varying, and with that also we get an understanding of how things are being related. You get it? And we can understand the differences also. So we'll get to know even more better when we go ahead and see the DNA fingerprinting process. So let's see that. So bulk DNA, satellite DNA is there. Satellite DNA is something that is repeated, and repetitive sequence, definitely we share some, and they also have some changes between us. So yes. Okay, let's go ahead. Let's analyze it first. So over here, suppose there was a crime site. Over here, a crime happened. And from this crime site, I got blood, hair follicle, saliva, skin. Either of these things. Suppose if I am getting anything from these sites, basically I get to understand the DNA. Yes. So basically, suppose I did a crime and I am telling you that I'm telling the police that I didn't do it. It was someone else who did the crime. It was Puchu who did the crime. No, Puchu does not make sense. Papu who did the crime. Okay. Okay. So the police said that okay, let us check. So the DNA which we got from here, it was taken, and you know what was done? a complementary strand of this particular DNA was being made, and it was tagged with some radioactive stuff. You now call this thing as a probe. What do you call this as? A probe. Probe is what? A complementary strand of DNA which is attached to a radioactive thing. Okay, this was the DNA of what? The criminal. Now, they had the doubt between me and Pappu, right? So, this is me and this is Pappu. First of all, the thing that is going to be done is our DNA are going to be isolated. So, isolation of DNA will be done, both of us. Second thing, both of our DNA are going to be cut by the restriction enzyme that was made to cut the DNA over here also. So, here also the DNA will be cut. So, anyway, so same restriction enzyme is used throughout. So, cleaving or cutting of the DNA strand with what? The restriction enzymes. After that, electrophoresis was being done. That means separation of these cut strands were being done. And you know what was being found? I'll tell you. Similarly, here also it was being performed. Okay, here also it was being done. So, this is criminal's one. It was found here, here and here. Okay, mine was also done. Mine was also here, here, here. Pappu's was here, here and here. When electrophoresis was done, they were ran and they looked like this. Now, can you directly check that if at all it's matching or not? No. I made it, that is why you are able to see it. In electrophoresis chamber, you cannot see it. So, basically, you need to transfer it onto a nitrocellulose sheet. Okay, so after this, you transfer it to a nitrocellulose sheet. You call this as southern blotting. After putting it on the sheet, you are going to run the probe. You are going to put the probe onto it. Now what is going to happen? The probe which is there, it will go and attach only to the area where it is complementary. So it came and attached over here. It came and attached over here. It came and attached over here also. In this particular case, it came and attached over here, and that's all. And that's all. Now, you view it after putting the probe. What do? You view it under some UV rays or autoradiography machine. What you will see? You will see that this and this basically matches. How did you get to understand that? Because this shows the radioactivity. You have attached probe. It is radioactivity, and you see the same pattern in the criminal. That means what? I am going to be the criminal. So why did it go and attach over there? I told you that repetitive sequences are found in all our organisms. Some of them can be same, but it's not going to be the exact way, exactly the way. So at some strand, yes, it can go and attach, but not on the other strands out there. And with the help of this DNA fingerprinting technique, you can find who did the crime, or basically, are we related? Mother and child relation and everything, because mother and child will be having maximum things being matched. You get it? So those can be identified. I'm not saying exactly everything will be matched, but maximum of the things are going to be matched. For that, you need to have either blood, hair follicle, the saliva or the skin. In the blood, if at all you are just taking RBC, red blood cell, that is not going to work. Erythrocytes and red blood cell does not contain nucleus. If there is no nucleus, no DNA. So RBC in the blood is of no use for now. Other cells are also there in the blood that can be used. So also understand that. Okay, that's all. So yes, with the help of repetitive sequence as well as the other thing out there, we were able to find out the criminal. Why? Because we are unique. We have it. Okay, we are all uniquely built. We also share some repetitive sequence, but it won't be exactly the same. Got it? So that is the whole thing that was given over here also. DNA fingerprinting process by Alex Jeffrey. Yes, here it was put down by Alex Jeffrey. So if at all you are asked about VNTR method, variable number of tandem repeats method, or DNA fingerprint method, everything, the answer would be the same itself. Okay, isolation of DNA. DNA is extracted from the sample, blood, hair, saliva, skin, thread, etc. Digestion by restriction enzyme, DNA is being cut. Separation by electrophoresis, transfer to the membrane, hybridization with the DNA probe, and visualization under the X-rays or whatever thing is there. So you will be seeing something like this. Okay. And that proves everything. Fine. So if at all you're given something like this and you're asked who is related, so basically this and this is related. This is not. Cool? So our DNA has bulk DNA and satellite DNA, small but unique repetitive sequence. This satellite DNA varies from person to person and helps in DNA fingerprinting. Since these sequences do not code for proteins, mutations in them do not affect health also. So repetitive sequence, any changes in them are not going to affect us. So my dear children, with that we complete molecular basis of inheritance. But if at all you have any doubt which is there, please put it down, and if at all you think that this video was helpful, definitely let us know about that also. Okay, wishing you all the best. Please take care. Bye bye.